Hi guys, I am Nisarga DC. My seminar topic is Lytic and Lysogenic cycle of viral infection. Actually, this is also known as a mode of replication. So now I am on to introduction. Before we talk about how do we grow virus and how can we purify them or even count them or enumerate them, we need to know what are different modes of replication in a virus. It is also known as a viral infection of host cell. So viruses do not follow just one mode. There are some who can follow different types of an replication cycle. There are certain viruses which can follow only one type of replication. So we will look into what are different modes of replication seen in the virus. What are the different steps in these replicative cycles and how the viruses bring about each of these steps. So firstly, what you need to know is that viruses can follow either lytic cycle of replication or the lysogenic cycle of replication. Now we move on to lytic cycle of replication. Okay. Lytic cycle of replication is also called as virulent because here viruses goes inside a host cell, multiply, replicate over there and then comes out. So in this cycle, viruses does not stay inside the virus, has no business inside the host. It's just going in, multiplying and coming out. So whereas in lysogenic cycle, the viruses has an option. The viruses has the option of going inside the host, staying inside the host for long as it wishes to and then coming out. So those the virus which follow such kind of life cycle where they get integrate with the host genome and stay inside the host. Such viruses are also called as a temporary viruses. So we have both virulent viruses. Sorry, we have virulent and like temporary viruses also, which means to say those that can follow only lytic cycle are those which have choice whether they want to stay integrated inside or they want to follow the lytic cycle and come outside. So before we talk about what are the those steps and what is the small difference between two types of cycle, let us look into broadly what are the different steps involved in replication. So those the steps that are involved in basic lytic cycle are what are shown over here. Lysogency, there is a small modification that occurs which I will be touching up later. Okay, so we move on to lytic cycle steps. So in the lytic cycle, first step is always the absorption, attachment, and followed by penetration, which basically means entry into the cell. Then it is replication or the biogenesis stage, followed by assemble of maturation and release. So these are the different steps involved in replication in a lytic cycle. Let us look into each of one in this detail. So, right, okay. So as you can see over here, there is a lytic cycle. We have the first step is attachment. Now in attachment, the viruses is going to get attached onto the surface of the host, or the viruses comes and binds to the surface of the host. It recognizes the receptor on the host cell, and once it recognizes that receptor, some particle on the surface of the viral cell will come and bind to the host cell. Now what I show here is of T4 bacteria, sorry. So that it is why you can see in the slide that is the mentioned as page or it is mentioned as a bacterial cell. But the lytic cycle is a similar for all the viruses, where in the first step is where viruses is going to come and recognize the receptor on the host and bind to the receptor. Initially, binding is always a reversible kind of binding. Here we call it as attachment. And then there is a covalent bond formation that takes place between the viral ligand or the viral surface and the host receptor. The irreversible binding is called as absorption. So the first step is always attachment and absorption to particular host cell This is followed by entry inside the host. Now, different viruses have different mechanism of entry. Some of them inject their DNA into the host. Some other they get engulfed into the host, and then inside they will go and get uncoated. Some other viruses are going to leave their capsid outside and the only genome enter inside. Some of them are enveloped viruses and some other viruses are going to leave their envelope outside and then nucleocapsid is come inside. So the mechanism of entry and penetration is different. What is shown here is for typho bacteria which injects its genome into the host cell. So now next step is penetration, which varies from one viruses to other. It basically it means the viruses has entered into the host cell, or at least the genome of the viruses has entered into the host cell. This is followed by the third step, that is the biogenesis. Actually, the replication part is gone here. Here, some multiple copies of viral components are synthesized. Now, unlike a bacterial or an animal or even a plant cell, a viral cell does not replicate by budding or fusion. So here viruses is none of their mechanism are going here, budding also or fusion also. A viral cell replication like a machinery means I have a 10 parts in me. I need to replicate each of those 10 parts, make a multiple hundred copies of each of those 10 parts, and then they will put together. So that is why the third step is biogenesis or replication, wherein all different parts of viral viron or the viral components are synthesized here. Multiple copies of genome, multiple copies of capsid protein, multiple copies of peplomers of the glycoprotein spikes, and any components that is there in a virus is synthesized in multiples. That is a biogenesis. This is followed by assemble and maturation. Now, the assemble and maturation can be actually considered as a one step where in this individual viral components are now assembled together. They are put together like how we can make machine by different companies like a I will have together and still they finally products come with means actual sorry exactly wait I'm getting sorry I will continue each sorry there are put together like how make a machine different companies synthesize it each part of the machinery and then one company is going to put it all together and sell the final product is same way here exactly the same way viral components are now put together. So the enzyme, the protein, the genome, all of it going to put together. It is going to be assembled and the viruses is going to get completed and fully developed, which we call as assemble and maturation step. When they have a lysis or the release step, now the release is the last part of the lytic cycle. As you can see over here, release is the last step. Now in case of release, the viruses is going to come out and of the host cell. This again viruses in mechanism. Some of them are extremely peace loving and some come out without disturbing the host cell. Some of them are highly destructive in nature. They break open the host cell and come out. So the release process is different in this diagram in which respect to the T4 bacteria, which is going to lysis into its host cell. So the ends release can be changed or the term lysis has been put, but it is not always the same case. Some organisms, some of the viruses do not lysis their host cell. They bud out of the host cell. So the mechanism of penetration and releases differ from one virus to other, but generally the steps are same. So now we have to move on to lysogenic cycle. Here, what happens is attachment and adsorption is same. Then the viral genome get inside, but the viral genome after getting inside of forming a viral component, or instead of getting into biosynthesis instead of that, it is undergo a step called integration. In integration, the viral genome gets integrated, it gets combined, it gets fused with the host genome. So here what is happening is that here viral genome which is shown in the blue is getting integrated with the host genome, which is in pink. Now, whenever the host cell divides into every progeny, the viral DNA will also be passed. So, if this cell has divided 100 times, I will have a 100 daughter cell of carrying the viral genome. When the time is right, basically when the conditions are not very favorable, or when the cell is where the host cell is going undergoing through some stress or some unfavorable condition. Under such condition, the viral genome will now come out, and the host genome, yes, come out of the host genome. This process where the viral genome is coming out of the host genome is called as an induction. So here, steps is also called as an integration. So these steps where it is coming out is called as also an induction. The stage where it is present as an integrated part of the host genome is called as a provirus stage. So if we are talking about page, it is called as a propage stage. If it is an animal virus, it is provirus stage, where the host genome is now integrated with viral genome. Now, as you can see here, the viral genome has come out, got integrated out of the host genome. It is will undergo rest of the lytic cycle, which means to say the biosynthesis, the assemble and maturation, and finally the release. So the lysogency is basically a cycle where the organism, where the viruses get to decide whether it wants to go inside, multiply and come out, or it wants to go inside, stay inside, after a while multiply and come out. That is the only choice temperate viruses have. So these are the two different types of viral cycle of viral infection. This is also known as a mode of replication in the viruses. Thank you so much. Thank you, sir.